For the last several weeks, I have had the opportunity to work in the April Hill lab and be a part of their vast research in freshwater sponges. I, along with my mentor Benjamin Cornegay and fellow mentee Melvin Noel, have been conducting an experiment on how BPA and BPS affect sponges. The reason why we use sponges is because of their similar genetic makeup to humans, as well as their easy use for experiments. Their similar genetic makeup makes them excellent subjects for comparing the effects of certain chemicals to human genes. The big question we were asking ourselves was how BPA and BPS affects the sponges. We were focusing not so much on the sponge, but the expression of the gene, which in our case was a gene called EZH2. For our hypothesis, we predicted that at a low concentration, the sponges would still be affected negatively. It was also predicted that BPS is just as harmful as BPA, or if that, even more. Bisphenol A is an industrial chemical used to manufacture polycarbonate plastics, canned goods, and epoxy resins. Exposures to BPA have high possibilities of health effects to the brain and behavior. Studies have shown that it has affected the early stage development of many living organisms. Bisphenol S is used as a substitute in BPA-free products. It also has the same chemical structure as BPA, but its harmful effects are not widely known by its consumers. We cannot start the experiment without extracting gemmules, which are the reproductive bud of the sponge. Within doing this, the sponges were cleaned with hydrogen peroxide and plated for growth. Next, we use serial dilutions to make our BPA and BPS solutions. Using the C1V1 equation, we calculated 160 parts per million of each chemical for this process, and 1600ths being the lowest, which is a very low concentration. Afterwards, we treated our sponges with the solutions, and two treatments were made. Then we needed to make cDNA, but in order to do that, we needed to make RNA from our treated sponges. Once this was done and we added our forward and reverse primers, we can now run a polymerase chain reaction, also known as a PCR. It shows a specific part of a gene, and for our project, we wanted to focus on the expression of EZH2. So we first tested EF1 alpha as our control. We also ran it on a gel electrophoresis to roughly show the amount of base pairs after the PCR. Due to the lack of time and insufficient supplies, we were not able to actually look at the expressions of the EZH2 gene. But after taking pictures under the microscope, we noticed something interesting. Some of the sponges had died because of the high level concentrations of BPA and BPS. All of the sponges who had received 160 parts per million were still in the beginning stage of growth. By comparing to our control, we saw that the lower the concentration was, the healthier the sponge was. So without looking at the expressions of the EZH2 gene, our data still supported our hypothesis.